Hi, I'm Colin McDonnell. For my final project, I designed a light programmable logic gate. I'm going to walk you through the 5 steps I used in designing this logic gate. First, I'm going to talk about using light as an input to genetic circuits. Next, I'm going to talk about ways of changing a genetic circuit post-assembly. Third, I'll talk about creating a universal logic gate that can be tweaked using the methods in step 2 in order to form any of the 4 major logic gates: AND, OR, NAND, and NOR. Next, I'll talk about how I modeled each of those components, and then lastly, I'll talk about the system-level behavior, and talk about some different research directions. So how do we actually use light as an input to a genetic circuit? Well, it's all based around the process of phosphorylation. Essentially, this response regulator protein turns off that promoter unless it's been phosphorylated, i.e. one of those blue dots has managed to bind with it, like so. Unfortunately, this process doesn't happen spontaneously. There are proteins called kinases that facilitate this interaction. The kinase shown here is special because it has a photoreceptor domain. That means that the ability of the kinase to do its job is actually dependent on the existence of outside light shining on the cell's surface. In this particular case, external light shining on the photoreceptor domain inhibits the kinase's ability to facilitate phosphorylation. This is what's called a two-component regulatory system. It's called two-component because there's one component outside the cell that's reading in external stimuli. That signal is transmitted inside the cell, or it actually impacts enzymatic behavior. Here, for convenience, I've implemented a quick inverter circuit. Now, instead of transcription being off when the light is on, and on when the light is off, the output protein is expressed when light is being shown on the surface of the cell. I've compressed this entire circuit into a simple shorthand of a light-activated promoter, shown here. I'll be using this throughout the rest of my presentation. Now I'm going to talk about a way of modifying genetic circuits post-assembly. That means that this entire process happens entirely within the cell. The key player here is the recombinase enzyme. This enzyme is able to perform site-specific recombination. Here's a simple circuit. Basically, the production of GFP is being regulated by an operator that's being bound to by repressor proteins floating around in the cell, represented by blue dots. Here's some recombinase enzyme, and these are the restriction sites that have been engineered onto this strand of DNA. The recombinase will bind and cleave the DNA at the 2 restriction sites, getting rid of the operator entirely. This makes the repressor proteins floating in the cell irrelevant. The last thing the recombinase does is ligate together those 2 pieces of protein to form a coherent whole. Now we have a constitutive promoter that's always transcribing GFP. There's some really nice things about using a recombinase to change the DNA strand inside the cell. First of all, it's unidirectional. After the recombinase chops out that operator, the operator won't be able to insert itself back into that strand of DNA ever. Second of all, it's permanent. Now that we have this new modified strand of DNA, the existing processes in the cell, like DNA replication, are working in our favor. This strand of DNA will be able to propagate long after the death of the cell it's currently in. In fact, it can propagate for multiple generations using the traditional methods of DNA replication. And lastly, the whole process is mediated by transcription. We can transcribe and translate recombinase proteins just like any other protein by just having its DNA in the cell and using the existing processes to see its expression. We know how to make arbitrary changes to DNA using recombinase enzymes. We also know how to initiate the transcription of a protein using a light-sensitive kinase. Here, we have 2 photosensitive promoters. One of them is sensitive to red light. The other one is sensitive to blue light. Certain photoreceptor domains from various species of cells are sensitive to different wavelengths. By finding one that's sensitive to red and another that's sensitive to blue, that enables us to make 2 arbitrary changes to a genetic circuit entirely through light-initiated transcription of a recombinase enzyme. We now need to come up with a template for all transcription-based logic gates that can be reduced using recombinases to any of the 4 major logic gates in use today. That's AND, OR, NAND, and NOR. We're going to assume some things right off the bat. First of all, the inputs to our logic gates will be inducers. Second of all, we'll have a constitutive production of R1 and R2, which bind to O1 and O2 respectively. Here's an example of an AND gate. In order to start transcribing GFP, both inducers need to be present, thus knocking the repressor proteins off of their respective operators. For the OR gate, we only need either I1 or I2 to be present. This is the NAND gate. Both inducers need to be present to start transcribing R3. R3, in turn, represses the output GFP, which means that GFP is only being produced if either I1 or I2 or neither is present in the cell. For the NOR gate, the presence of either inducer begins transcription of R3. That then inhibits the output GFP. So GFP is only being produced here if neither inducer 1 nor inducer 2 is actually present in the cell. This set of 3 DNA strands can be reduced down to the full set of 4 logic gates that I just showed you, using the action of only 2 different recombinases. Even though the top 2 strands look the same, their recombination sites on the top and bottom strand are slightly different for the blue recombinase. Let me prove to you that we can make any of the 4 previous logic gates out of these 3 strands of DNA, using precise application of recombinase enzyme. If we add no enzyme whatsoever, these circuits actually act as the AND gate described before. As you can see, there's a promoter immediately upstream from R3. That means that that repressor is being continuously produced. Thus, the GFP in the bottom strand is not being produced whatsoever. So we can ignore R3 and the entire bottom strand of DNA and just look at the top strand, which is then operator 1, operator 2, followed by GFP, and that's going to have the exact same behavior as the AND gate shown to the right. Here's the OR gate. In this situation, we apply just the blue recombinase. That means that we chop out operator 2 in the top strand and operator 1 in the bottom strand. What we're left with is the same as this OR gate here. We can still ignore the bottom strand of DNA and both R3s on the right side of the top 2 strands. Because we've chopped out O2 from the top strand and O1 from the middle strand, we're left with a circuit that looks identical to the OR gate to the right. Here again for the NAND gate. If we apply the red recombinase, we chop out the GFP and the constitutive promoter promoting R3 from both the top and middle strands. Thus, what we're left with is exactly the NAND gate I've shown you before. And finally, here's the NOR gate. If we apply both recombinases, then for the top strand, all we have left is operator 1 and R3. For the middle strand, all we have is operator 2 and R3, and the bottom strand remains the same. This is the same as the NOR gate I've shown you before. Thus, by applying some combination of our 2 recombinases, we can go from the general template genetic circuit on the left and produce any of the 4 logic gates I described before. Now I'll discuss how I went about modeling all the parts of this system. I've split up the system here into inputs, intermediates, and outputs. The only input to the system is light. Light directly modulates the transcription of the recombinase, according to the relationship I have here. This relationship was first described in Tabor et al. In 2009. The difference between my model and Tabor's is that I'm assuming there's no leakage in the system. That means that the B-min value is 0. Thus, when the amount of light is 0, the amount of mRNA produced is also 0. However, as the amount of light approaches infinity, the amount of mRNA produced approaches Bmax. A reasonable value for Bmax, as drawn from Tabor's 2009 paper, is approximately 5 nanomolar per second. Moving further to the right, we see that the mRNA recombinase has some constant degradation rate, but we also see it being translated into recombinase itself. That recombinase also has some predetermined degradation rate. It can also combine with our template and DNA to produce the other 3 types. This box diagram doesn't completely represent the full complexity of modeling this circuit. There are in fact 2 forms of light, 2 different recombinase mRNAs, 2 different recombinase proteins, and multiple variants of the OR DNA, NAND DNA, and NOR DNA. Here are the results of my simulation. On the left, you see the behavior of the system given a single spike of red light at time equals 100. Immediately, the AND DNA begins degrading, as the integrase surges into the system and begins transforming it into NAND DNA. Over time, the amount of NAND DNA approaches the original amount of AND DNA, which is 1 nanomolar. On the right, you see a similar picture, though for a pulse of blue light. Here, instead of the production of NAND, we see the production of OR. Here's a more complex simulation. Here, we have a spike of red light at time 200 and a spike of blue light at time 500. At first, we see the AND DNA beginning to drop off as we see the rise of NAND DNA. However, once the blue light pulse hits at time equals 500, then both integrases are present in the system. That means that both NAND and OR DNA are rapidly being converted into NOR DNA, which results after both recombinases have successfully excised their respective pieces of DNA. On the right, we see a longer-term vision of the system that shows clearly that with 2 pulses of light, the amount of NOR DNA slowly approaches 1 nanomolar. That is, all DNA in the system eventually gets converted to NOR DNA. Now I'll describe the system-level behavior. What we want to be able to do is insert 3 template DNA strands into a cell, and then determine which type of logic gate that forms into by modulating it with pulses of light. Here are the steps to do so. Add all 3 of those distinct DNA strands, as shown in the slide on the logic gate template, into a cell. Make sure that all the recombination sites match exactly with the diagrams shown here. Add in plasmids with the light-sensitive promoters, encoding 2 orthogonal recombinases. These plasmids encode the light-sensitive kinase that's used to actually enable photosensitivity in the system. We also need to constitutively produce certain things for this circuit to work. First, we need to produce R1 and R2 to enable the function of the logic gates. If you remember, that was one of our assumptions in analyzing the logic gates initially. This is what enables us to use inducers as the inputs. Second, we need to constitutively produce that photoreceptive kinase protein. That's what actually enables us to have this circuit respond to the presence of light. The fourth step is actually shining the appropriate combination of light to bring about the desired behavior. Here's a simple key. No pulse results in an AND gate. Blue results in OR. A red pulse results in NAND, and both result in NOR. Now we can use inducer inputs to perform transcriptional logic using our custom logic gate that's been determined by our choice of pulses of light. The dynamics of transcriptional logic are already well understood, which is why I chose not to model it in this system. This project is the first step towards enabling more complex behavior in genetic circuits that's modulated entirely using light. One can imagine a light programmable gate array that consists of multiple logic gates all hooked together in series, each of which is changed at the DNA level using a different frequency of light. You can also bring about more complex spatial behavior by using photomasks to determine which cells get light and which cells don't. Long term, we can imagine cells that can be rapidly reprogrammed to perform complex behaviors entirely using light.